E, and N antigens are put in. Some lipid membranes or scaffoldings are also added. And then this assembly of antigens into a virus-like particle is going to happen in vivo. And again, the antigens are going to be presented vertically or in the 3D shape, which stimulates good immunity. This is still not present in India, and there are no commercial trials as yet. Now, moving on to the one, again, so there's a lot of confusion as to what this vaccine is. Is it good? Is it bad? So let's just understand what the vaccine is. The messenger RNA vaccine, which is being marketed by Moderna as mRNA 1273. The RNA virus produces messenger RNA, which further goes down to encode proteins. This is nothing new. This is what happens in all cells, whether it be viral or be human. Now, this messenger RNA is taken to code for the S protein. But question is how to put in this messenger RNA inside the human cell. If we leave it free floating in the bloodstream, it will be degraded and it will never enter the cytoplasm. Therefore, lipid nanoparticles are going to be used as carriers and protectors of the messenger RNA. And once it goes in, it will have two target cells. One will be the muscle cell or the myocyte, and the other would be the antigen presenting cells into the first draining lymph node. Now, inside the myocytes, the outer lipid layer of the nanoparticle is going to fuse with the plasma membrane. This messenger RNA is going to enter the cytoplasm, and just like any other protein which is coded by messenger RNA, this S1 protein will start getting coded, expressed on the myocyte, and will start acting as an antigen, and slow immune response will develop against it, and eventually both cell-mediated and antibody-mediated immunity will result. Secondly, if it manages to enter the antigen-presenting cell in lymph node, then the response against it would be much faster. The immune-competent cells will work via MHC2 pathways, stimulate T helper cells, and finally a rapid immune response will develop. Now, all is good in theory, but the operational problems are that this messenger RNA vaccine, to remain viable, it requires a storage at -20 to -70 degrees centigrade. Now, as you know, even in peripheral parts of India, it's tough to get even refrigerators. So it's very far cry to think whether -20 and -70 degrees centigrade deep freezers will be available. So I don't see this particular vaccine coming in a big way in India, but let's watch out for its efficacy. Now, coming to the DNA vaccine, if you remember, I told you there are two choices. One is to leave the DNA free floating in the blood, but there the problem is how will it enter into the cell. So that is the main problem with the Inovio vaccine, that the efficient way of making this DNA enter into human cells is not easy. So the vaccine is still not coming in trial phase, not because the DNA vaccine is difficult to produce, but we are still working on the delivery system of this vaccine. So one method is something called electroporation, that you give an injection of this vaccine and after that apply a slight electric current. Now don't be alarmed. The DNA is negatively charged. The cell surface is also negatively charged. So the DNA does not have any natural affinity for the cell. So if you create a localized electrolyte change or something is there by applying a very mild electric current, you might be able to create a situation in which the DNA, which is negatively charged, enters the cell and then is taken up like any other antigen. Viral vectored vaccines, this is the one that we are talking about in India, is Covishield. So that is the DNA we have. It's not good to leave it free. It requires a shell. So that shell can be a replicating viral vector or a non-replicating. But as I told you, if a live vector is involved, a fast-track vaccine cannot be produced because safety is a concern. So currently only non-replicating vectors are being considered, and adenovirus is one such vector. So chimpanzee adenovirus is being used by the AstraZeneca vaccine, and the Sputnik 5 Russian vaccine is using a human adenovirus. So the chimpanzee adenovirus is modified so that it cannot cause disease. The SARS-CoV-2 DNA is put inside it, and theoretically, if it is given, the adenovirus infects the nucleus of human cells and causes a natural kind of infection wherein the antigen produces the S1 antigen and natural immunity results. So this is the AstraZeneca vaccine. India is partnering in this trial. You have to remember that all the DNA or RNA being discussed in these slides for India will result from the Indian strain of the vaccine. So to compare and address concerns, live attenuated viruses have the maximum antigenicity but substantial safety concerns. That is why they are not being put in fast track technologies. So if you inactivate the virus, the concerns become very low. But for us, a company like Bharat Biotech, which is so experienced in producing and distributing inactivated vaccines, is something to be seen in a good light. And logistics of distribution are very good because the temperature required for the vaccine is doable. Non-replicating DNA viruses have a high safety margin. They can be produced highly, but we have to look into the distribution in India, and with the political will, this can be done. DNA plasmids, it's difficult to circulate them and put in by electroporation. And soluble nanoparticles and all, they have a long way to go. So to summarize, the SARS-CoV-2 virus envelope spike protein is chosen as the vaccine target. Inactivated vaccines will have all antigens. Recombinant proteins can be assembled into simple S proteins or virus-like particles. If you're using the genetic material, RNA or DNA can be used. If RNA is being used, then it has to be put into a lipid nanoparticle. If DNA is being used, it can be given in free DNA or inside a viral vector. So at the end, I would like to just point out the privilege that we all have for being in India, where the vaccine strain is indigenous. It is definitely going to target Indian antigens. The technology for producing this and distributing this is present in India. So the choice is yours whether you take the vaccine or not. But you should know which are the options available and to choose with a good knowledge about the options. Thank you so much.
Thank you so much, Viri. I believe that was good and insight into the type of vaccines which are important, and as a part of microbiology, that was something which we really wish to understand. Now, when we are here, when we know about different type of vaccines, now the question arises that there are a lot of concerns about the clinical trials and how these clinical trials are conducted. So we have been hearing a lot from media that there is a kind of emergency use authorization for these vaccines. But as Viri has very nicely mentioned, that for the view of live virus vaccine, usually it is not done and any process is not cut short. So we would like to invite now my friend Jaykaran from Department of Pharmacology to have a brief overview of this vaccine development, and I request him to be quite specific about how it happened in this case of Covishield and Covaxin. So over to Jaykaran.
Good morning, everyone. Now, in coming slides, I will be talking about the overview of vaccine development in very brief. So basically, every vaccine candidate has to undergo these steps before it gets approved for use in humans. And these steps can be divided into two broader categories, preclinical and clinical. Now, in preclinical research is basically done in the laboratories, academic research, part of academic research, basic research. And in clinical phase, the vaccine candidate tried in the humans. And after phase 3 trial, the approval is given by the drug regulators. In the case of India, the drug regulators are Drug Control General of India under the CDSCO, which is Central Drug Standard Control Organization. And even after getting the approval, the vaccine candidate is actually being analyzed for the side effects which are not so common, as well as if there is any interaction with other drugs, or maybe tested for the pharmacoeconomical or pharmacoepidemiological data. So it's a time-consuming process. Usually it takes 1 to 2 decades to get one candidate out of many which start at this phase, and it requires a lot of resources in terms of skill, money, and time. So in the case of preclinical stage, the aim is to find the vaccine antigen. It may be a protein, it may be some genetic material, and in the case of COVID-19 vaccine, Chinese scientists published the genome data of this virus in January itself, and within 2-3 months, Moderna and Pfizer they could find the specific antigens. Now, these pharma companies, they have many antigens in mind, and they also have backup antigens. If first one or two antigens get failed in the research, these people may start working on some other antigens. So this is the most important part at the early stage, preclinical stage, where the identification of antigen happens. Then animal studies are done to evaluate the safety and efficacy. Once the vaccine candidate clears preclinical stage, it goes to clinical stage, and in every stage, permission is sought from the drug regulator. Now, phase 1 stage, sometimes divided into phase 1 and 1A and 1B. Now, this is first in man study, first in human study, and in the phase 1, healthy adults are included, or healthy volunteers are included, and this phase has very less sample size, less than 100, and immunocompromised patients are not included. Immunocompromised volunteers are not included because there are chances of more side effects in such kind of subjects. Then multiple phase 1 trials are done. The subsequent phase 1 trials are known as phase 1B trials, where different populations are used, or different vaccine candidates are used. Sometimes at early stage, the pharma company tried different kind of adjuvants. So even that can be tested in the phase 1 studies. And now if you look at the objectives, so the primary objective for any phase 1 clinical trial is safety. In the case of vaccine, it is safety, reactogenicity, and of course pharmacokinetics. While attempts can be made to see the immune response also. So phase 1 trials are usually open label trials, non-randomized trial, but in the case of vaccine trial, even randomization may be done, even control group may be there at later part of the phase 1 trials. So once the vaccine candidate clears the phase 1 trial, it goes to phase 2 trial, and in this case we have many subjects. Now in the case of drugs we have patients, but in the case of vaccine we will have the healthy subjects, and it may have large sample size, maybe few hundreds to even 1,000, and it may have heterogeneous population, maybe having different age groups, maybe having different population. And the primary objective of phase 2 trials are safety plus finding the adequate dose, finding adequate dose schedule, and vaccine preparation, pharmacokinetics. And secondary objective is also to see the efficacy and immunogenicity. So sometimes phase 2 trial is divided into phase 2A and phase 2B. So phase 2A trial are also known as proof of concept studies, where it generates good amount of data related to efficacy of the intervention. In this case, it will be the vaccine. Phase 2B trial is mainly responsible for finding the therapeutic dose, range of therapeutic dose, effective dose, toxic dose. So sometimes drug approval may be sought even after getting the phase 2A trial. It was done in past, where few drugs like bedaquiline was approved just after phase 2A trial. These trials are having more power because it has more sample size. Randomization is usually done in these trials. Even blinding can be done, and comparison can be done with the placebo or active control. And of course, we need to have multiple phase 2 studies. Even like phase 1 studies also, multiple trials need to be done, maybe simultaneously, maybe different part of the world, in different geographic location, in different populations. Then after completion of phase 2 trial, it goes to phase 3 trial, which is a trial having larger sample size. Thousands of target subjects are included in such trials. The objective is to confirm the efficacy and safety, and these trials may have superiority trial design when no other intervention is available. it may have non-equivalence, it may have equivalence of non-inferiority design when other candidates are available. But these trials are very large trials, and these trials are needed for regulatory approval. So again, a pharma company may have to do multiple phase 3 trials before it get approved. And after getting the data from phase 3 trials, data are submitted to the pharma, submitted to drug regulators. So these drug regulators have subject expert committee, which has research methodologists, statistician, as well as clinicians and administrators. And based on the data submitted to them, the decision is taken. So after approval, to find the rare adverse effect, phase 4 trials are done, and this is done after the approval of the vaccine candidate, and it can be this candidate can also be tested for pharmacoeconomics, for pharmacoepidemiological studies, for drug interaction, for different routes also. So everything will come in the phase 4 trials. Now, what is emergency use authorization? So, majority of drugs approved till now for the COVID-19 are through this route only, emergency use authorization. Now, this emergency use authorization, this term is present in the FDA documents, but such term is not available in Indian context. In our new drug and clinical trial rules, this term is not there. We have some other term known as accelerated approval. So there are some conditions which, as per the FDA document, there are some conditions where emergency use authorization is permitted, and these conditions are: the disease is serious, life-threatening, and there are some evidence of effectiveness. So the specific word is used in the document, may be effective. So it means that the people, the subject expert committee people are ready to accept even low level of clinical evidence. Then risk benefit ratio should favor the benefit, and if there is no alternative exist to treat or prevent that particular disease. So a totality of evidence is considered based on the data submitted to the regulatory authorities. Decision is taken whether to give the emergency use authorization or not. Now, for the COVID-19 vaccine, FDA has very, very objective criteria. And if you look at the published trials, you will see that these trials has follow this FDA criteria. The first criteria is that it should be phase 3 data. No, we are talking about the FDA criteria. So there should be a phase 3 data, and even interim analysis will do. Or if a phase 3 trial is prematurely stopped because of getting early benefits, even that data will do. So there should be phase 3 data, and median follow-up for safety in these phase 3 trials should be at least 2 months. So if half of the subjects included in these trials follows up for 2 months, then this will be satisfied. So this is the second criteria. And the third criteria is, for safety estimate, there should be at least data of 3,000 subjects. So all three should be followed, all three should be fulfilled. So after fulfilling all these three criteria, emergency use authorization can be given. Now, FDA has very categorically mentioned that when these vaccines are given, all those who want to take the vaccine, want to get the vaccine, they should be told that these vaccines are approved through the emergency use authorization, not through the normal process, and it's purely voluntarily to get the vaccine. In the case of India, Indian law says that we have a specific term called accelerated approval. Now, that is given in the case of serious or life-threatening conditions, whenever there is a unmet medical need, and for that we have these two very important criteria: that good efficacy should be observed in the phase 2 trial. Even in the phase 2 trial, we have even if we do not have phase 3 trial, if we have data of phase 2 trial, then also accelerated approval can be given. Or we have phase 3 data and we do not have clinical endpoints. We do not have hard endpoints. Even we are having surrogate endpoints. In case of vaccine, we may not have actual data of prevention of infection, but we may have data of seroconversion. So even based on that kind of data, accelerated approval can be given. So this is as per the Indian law, which is very categorically mentioned in the documents, new drugs and clinical trial rules. So we need to understand that vaccine, any intervention take years to develop, but in the case of vaccine, it was done very fast. Within 2 years, we could produce the vaccine without compromising the safety, because a lot of efforts were done by the governments, by the NGOs, by the pharma companies, by the universities. This was a Herculean task, and this has changed everything related to vaccine development. And in future, we believe that because of this experience, we are going to develop more and more vaccine in the short time. Thank you.
Thank you, Jagaran. So that was really nice to get this idea on what is the difference between the accelerated approval and emergency use authorization. As very nicely mentioned by him, that it is actually the FDA terminology which is being used across countries, but in India we call it as accelerated approvals. But still we are getting these kind of words in media and in most of the reports shared by even government agencies. Now, we are towards the main part of this actual part, and that is about when we are talking and when we are hearing that we are going to get Covishield on 16 January. So let me take this opportunity to invite Dr. Manoj Gupta from my own department, to say in brief what we know about Covishield. Are there some studies published in terms of animal studies or human data? And what is the rate or how it's going to be administered? So over to Manoj for Covishield.
Good morning, everyone. So the first vaccine, Covishield, which we are going to introduce or inoculate in our country, developed by Oxford and AstraZeneca, and Serum Institute of India got the license to manufacture in India. So I'll be going through the published literature first. What are the different trials? What are the findings? The safety profile, immunogenicity profile, efficacy profile. So first study which was published first trial which was done on animals, it was published in Nature in October 2020. So this is basically adenovirus vector-based vaccine, and this is a generic name CHAdOX1 and COP19, which encodes the spike protein of SARS virus, SARS-CoV-2. And this was found immunogenic in mice and could elicit a robust humoral as well as cell-mediated immune response. It induced a balanced humoral and cell-mediated response in rhesus monkeys, and both for T helper cells, basically. A significantly reduced viral load was also observed in bronchial alveolar lavage fluid and in lower respiratory tract tissues of vaccinated rhesus monkeys, and no pneumonia was observed in vaccinated SARS-CoV-2 infected monkeys. However, there was no difference in nasal shadings between vaccinated and controlled SARS-CoV-2 infected monkeys. So this was the animal studies. Then the phase 1 and 2 trials, they were combined together for this particular vaccine. It was published in Lancet in November 2020. It was done in 5 different sites in UK, and the same vaccine, which was chimpanzee adenovirus vector vaccine. The basic science behind this has already been explained by Dr. Vidhi, which was expressing SARS-CoV-2 spike protein, was used, and healthy adults of 18 to 55 years were enrolled in the study. Safety was assessed after 28 days of vaccination, and the final result of these 2 trials showed that it has an acceptable safety profile, and homologous boosting increased antibody response was observed. Later on, phase 2 and 3 trials were combined together. Another study published in Lancet in December 2020, and they observed that they enrolled healthy adults again, more than 18 years of age, but this time they had taken age escalation manner, or we can say immunogenicity subgroups were taken. So 3 categories of age groups were made: 18 to 55 years, 56 to 69 years, and 70 years and older. And they observed local reactions as well as systemic reactions. So the interesting finding here was as the age was increasing, local reactions as well as systemic reactions, or we can say reactogenicity, was decreasing. So finally, they concluded that it's better tolerated in older adults than in younger adults, and found with similar immunogenicity across all age groups after 28 days of boost dose. Most common local reactions which were observed were pain site injection site redness, itching, induration, and most common systemic reaction was fever followed by headache, malaise, joint pain. But they could be managed with paracetamol. Another study, very interesting study, which was published on large data of phase 3 trial in Lancet in December itself. So this was a preliminary analysis of ongoing trial, which was done in different countries with different geography in UK, Brazil, and South Africa. A total of 23,848 participants were enrolled in the study. Again, the criteria was same. All were more than 18 years of age. In this particular study, they proved that if we use two standard doses 1 month apart, then the efficacy is 62.1%. And another, this was basically 3-arm study. In second arm, they used low dose followed by standard dose. So they make half dose first and then full dose. So efficacy was 90%, and the overall efficacy was 70.4%, which is usually claimed by the manufacturers. So in India, we are following this 2 standard dose pattern, not this low dose and standard dose. There are 2 reasons. First is that this trial was extended into India, and we have done this with 2 standard doses only. So this is not done in India with low dose. Second thing is it has wider confidence interval. It's 90% efficacy. So authors themselves has discussed that this should be further explored and with a large sample size or with a large clinical trial. So that's why this 90% efficacy or low dose followed by standard dose is not followed in our country. We are following this 2 standard dose pattern. And as far as the safety is concerned, we can say that after 21 days of the first dose, 10 cases were hospitalized. As per the literature, all 10 cases were in the control arm. And out of those 10, 2 were severe COVID-19 and 1 death was reported. But everybody, all 10 were in control arm only. There were many adverse events also, severe adverse event, 168 severe adverse events were reported. But only 3 were classified which were possibly related to vaccine. They have sent them for further investigations. These severe adverse events were high-grade fever, which could be managed in 1 or 2 days, but they require hospitalization. But interestingly, there was no pattern in the events or adverse event that provided a safety signal in the study. So trial was continued. Then this trial was enrolled in India. So some 1,600 healthy adults have been enrolled, 400 for immunogenicity cohort and 1,200 in safety cohort. So Serum Institute of India, along with ICMR, they are doing this trial together. Then they submitted the report to DGCI of India, and though this 1,600 data is not available in public domain, but they have submitted the interim safety and immunogenicity report to DGCI, and based on that, they have given the restricted use in emergency, the permission to use this vaccine in emergency situation, the criteria for which has already been discussed with Dr. Jayakaran. Recently, this price has been fixed. Government has given a purchase order to Serum Institute of India. The price has been fixed as 200 rupees per dose and 10 rupees as GST. So it's 210 rupees per dose. Some 1.1 crore doses has been purchased by government, and yesterday it was shipped across different major cities of the country. As per the media reports, the cost for private use or in the market will be 1,000 rupees, but it's not confirmed yet. And Serum Institute has stockpiles and over 50 million doses. They are saying 55 or 60 million doses they have reserved for the country. If you look at the stages of inoculation, in stage 1, in the initial phase, some 30 crore beneficiaries has been targeted in 4 stages. In stage 1, all the healthcare workers will be targeted. So 1 crore population approximately. Then in the stage 2, frontline workers, municipal workers, disaster management authorities, those will be targeted, some 2 crore population. Government is planning to give them those 3 crore free of cost, and probably Covishield will be used in those initial 2 stages. Then in stage 3, by the time the data for Covaxin will come, and then we'll move to stage 3 in which citizens with comorbidities will be targeted, irrespective of the age group. And in stage 4, 50 years and above people will be targeted. Again, they have been divided into 2 groups, 50 to 59 and more than 60 and above. So 60 and above will be targeted first, and then 50 to 59 will be targeted. So these will be stages of inoculation. If we briefly look about the vaccination details, it has 2 doses, which should be given 4 weeks apart, which we are following here, which we will be following here, and it should be given intramuscularly. The best thing is it can be stored in normal temperature 2 to 8 degrees Celsius. So we already have a very big immunization program. So we are all maintaining this temperature for many vaccines. So it will not be very difficult for our country to transport this vaccine even for interiors like Pfizer or Moderna, which require very low temperature, minus 20 to minus 70 degrees Celsius. This is all about Covishield vaccine. Now, over to.
Okay, thank you, Manoj. So I believe after this, having the kind of description about the Covishield, it's the time to know something about Covaxin, because you must have heard the recent report by Government of India that the states are not going to get any choice to have between Covishield and Covaxin. So we never know which vaccine we are going to get. So let's have a look on Covaxin. So over to Dr. Akhil Dinesh Goyal from my own department.
Good morning, everybody. So let's quickly go through the evidence about Covaxin. So I'll be going through Covaxin under these headings: animal study, phase 1 and 2, and recent updates. So the animal study results have been published as a preprint in Nature Research, in which they have found that what they did was a whole cell inactivated vaccine were administered in 20 monkeys in 4 groups. Group 1 was placebo, and other groups were the vaccine with various doses and adjuvant combinations. So on day 0 and day 14, the vaccine was given. And on day 28, the monkeys were exposed to a virus challenge. And then from days 1 to 7, after the virus challenge, there are various nasal swabs, throat swabs, bowel fluid, urine samples, and others were checked for the presence of virus. And on day 7, the animal was sacrificed and the histopathological examination of various body tissues was done for the presence of COVID. The results showed protective efficacy in terms of IgG and neutralizing antibodies. Also, there was reduction in replication of virus in the nasal cavity, throat, and lung tissues. There was no evidence of pneumonia as seen on histopathology, and no adverse events were seen in the animal group. The phase 1 study also has been published as a preprint. This was a double-blind multi-center study in 375 healthy volunteers, again divided into 4 groups, 1 placebo and 3 other groups having various doses of the active vaccine and adjuvant combinations. In this, the main side effect which was seen was local reaction in the form of pain after the first and second doses. Among systemic reactions, most common seen was headache in the 6 microgram group, followed by fever and fatigue after first and second doses both. No major side effect was seen in the phase 1 trial. In phase 2 trial, again, this is a double-blind multi-center study in 380 volunteers. This was not a placebo-controlled trial. We have only 2 groups. One group with 3 microgram dosage and the second group with 6 microgram dosage given on day 0 and day 28, and immunogenicity analyzed on days 42 and days 56. In phase 2 trial, it was seen that after the dose 1 given on day 0, there was a 4-fold increase, more than 4-fold increase in the neutralizing antibodies on day 28, and which further increased on days 42 and 56. However, the seroconversion was not 100%. Almost 50% seroconversion can be seen by the 6 microgram dose on day 28, which increased to 96 and 98% on days 42 and 56. Again, the main reactions, adverse events which were seen was in the form of pain, both after dose 1 and dose 2, seen in around 3% of candidates. And the major systemic reactions which were seen was malaise, headache, and fever. The total number of adverse events were between 8 to 10% both after first and second doses. The phase 3 trial in 25,000 volunteers is underway. The interim results are likely to come by end of this month or next month. In summary, both Covishield and Covaxin are given in 2 doses. Their storage is around 2 to 8 degrees centigrade. And the price is approximately 220 for Covishield and around 300 for Covaxin. Thank you.
Thank you, Akhil. So I believe now, as we know that now we have some baseline knowledge about the process, the procedures, the authorization from the regulatory authorities. Now it's time to know how we are prepared or how much we are prepared for these vaccines at our own place, at our own institution. So just to share with you, as you all know that the process was started a bit before in year 2020, and it was on November 6th, I believe. You must have noticed there was an email sent to all of you, and through the Google form where every one of us is requested to give their important details, which are to be shared with Government of India to have a database of healthcare worker for prioritization to receive COVID-19 vaccine. Believe me, we get the responses from many of us, and I would like to share with you all is that as per the Government of India guidelines, the prior registration is mandatory for this vaccination. So there would not be any spot registration by now, and we have been informed by the ministry that the data which has already been shared with the Government of India is already shared with our partner agency of UNDP, where they have already freezed the entire data. So once the data is freezed or it's frozen, what we call it as, the vaccination is going to be started. There is a software which is available by the name of COVID software. COVID, it stands for COVID-19. It's a very intelligence network which we are already using in our different vaccination programs. So through this COVID software, our details would be there available to government. And we'll get this kind of sheet. It's a glimpse or a snapshot of that process. You can have a look on that session summary. So you'll get this kind of message, which is a message for you to come for the vaccination at AIIMS. Today is a dry run, and we are going to see if there are any hitches or glitches to rectify when we have a final process on 16th of January. Just have a look on our site. It is now currently by getting the necessary approval from Director Sir, we have established it in the first floor of OPD block. So there are two blocks in that. Now in B block and in A block. Now there are 4 such sites as per Government of India instructions. At one site in one day, not more than 100 beneficiaries can receive this vaccine. So once this message is there, we need to come to these 3 rooms which are there. Now, one in the B block where there is a medicine OPD and adult vaccination OPD, and one in the A block where there is a dermatology OPD and gastro-medicine OPD. So this is a kind of arrangement which is there. The first room, which is the registration room, where there is a verification of the details which are shared with Government of India. The second room would be the room where the vaccine is going to be administered. And the third room is the observation room, where we need to sit for minimum 30 minutes. So it's a request to all of you that whosoever receives the vaccine, we need to be there in that room for a period of 30 minutes under observation, and for having any AFI. If unfortunately we get any AFI, it needs to be reported and to be managed. So this is my last slide before we move to the questions. So on 13, today is 13, so we are going to have a dry run in our own institution. So it's going to be started on 16th of January. And within a period of 4 to 5 days, we are hoping to cover the entire registered beneficiaries who have so far been registered from our own institution. So remember, there are 4-5 points just to summarize that though we have this vaccine with us, but we still need to follow the COVID appropriate behavior because the pandemic is not still over. So that appropriate behavior we all know is SMS, which is the social distancing, the mask, and the last but not the least is sanitization or hand hygiene. Second one, which is very important, is the completion of scheduled doses. As we all know that these are 2 doses which are to be administered. It's safe, you know, by the presentations today that there are a lot of data available in public domain. It's been peer reviewed, it's been published that the vaccine is safe. We all know that vaccine is important to not for us, but to build the herd immunity, because in natural infection we never know that how it's going to wane off. But in through vaccine we know that it's been studied well that there is a sustainable herd immunity with the help of vaccine. And last but not the least is that even if we are not saved from the attack of COVID-19 disease, even after getting a vaccine, the studies have shown that there would certainly be less severity if any one of us who received vaccine unfortunately got the spell of COVID-19. Thank you so much for hearing us, and I believe now is the time to take questions. So we have a lot of questions. So we have a question, and we'll shift questions to our colleagues who are here. So the first question we received from Abhishek is: has chimpanzee adenovirus been used as vaccine vector in any disease for mass vaccination? So
No, sir, it has not been used for mass vaccination. However, it has been used as a therapeutic strategy for patients of ADA, adenosine deaminase. So, no mass vaccination has been attempted as yet.
okay, so we have a next question. What about the AstraZeneca drug, where it was reported that low second dose was not actually part of trial and got administered by mistake? How reliable are results? Actually, we have all these news. We went through this, Dr. Madhubi, and there is no content in these news. We did see all those reports available in the social media, but there is no content. These are only news which have been reported through different agencies. So there is one more question. Is phase 3 trial being completed for Covaxin?
So phase 3 trial is underway. It started in December 2020. No interim results are currently available. But as per the Indian laws, even after phase 2 trial, if the safety data is available, accelerated use permission can be given by the DGCI.
Yeah, Dr. Neeraj is also asking a similar question, that do we have any interim data for phase 3 for Covaxin trial? And I believe we've already answered, so it's not yet available. Then one more question: why US is not believing on the results of Covishield from UK? There is no claim that the data is not clean, though the results are published in reputed journal Lancet. What is the truth? It's about Covishield, they're asking.
I don't have a certified answer to this, but if you ask me, I think it's about the money. Covishield and Covaxin and others are being put together, and there's a race to reach the first. So everybody who can produce a good vaccine is producing, and the others who have extra time are just publishing negative literature about the others. So let's wait for the actual results to come before taking a decision.
Thank you, Vidhi. So we have one more question from Dr. Bhatnagar, and she's asking how long the effect of vaccines last.
We don't know.
So we never know, because this is something which we call as the long effectiveness trial. Actually, it's a new vaccine, so we never know. And that can only be established by way of antibody titers, which will be done later on. So Bharat Paliwal, Dr. Bharat Paliwal is asking about, first of all, thanks. We really acknowledge your thanks. And now he's asking about that those who have already got infection, should they receive any kind of vaccine or not? So, currently it's been said that we already know from our own experience that those who have already got a spell of infection, either they got reinfection or there is a waning immunity. So antibody titers are not being maintained. So it's been proposed that yes, those of us who have already been having a spell of COVID-19, they are actually suggested to take this vaccine also. Pradeep is asking, Shobhan is asking how the injection site reaction is different in terms of incidence and severity from the existing vaccine. So do we have any answer? It's similar actually. So the data, actually there is a leaflet, I believe somebody has also posted in a common group. It needs to be shared with the beneficiary. So we'll also share the soft copy with all of you. The data says that it's the similar one. So Dr. Sharma is asking, is there any data regarding developing autoimmunity reactions like asthma, renal, dermatological, or joint pain or cardiomyopathy? So I think it's not yet, because these kind of data is not, because it's not been reported. So we are believing on the data that no such reactions or these adverse events were there. Indian efficacy data for Covishield, how sufficient and reliable, Dr. Didil, Dr. Sierra? Not in public domain, but actually the thing is that there are questions related to Covishield even. They are saying that this is the Serum Institute of India's vaccine which we are going to get, and the data which is available in the domain is from AstraZeneca. But the kind of technology which we are using is the similar one, and so I think it's not a point of much worry. So I believe most of the questions are done. No one, Dr. Pradeep Dwivedi's question is also there. Covaxin vaccination will be done in clinical trial mode. Any idea? Dr. Dwivedi is asking.
It has been approved after phase 2 trial, and there's nothing like a clinical trial mode.
That's true. This is nothing like a clinical trial mode, though there are some reports which are saying because the third phase is not there, but no, it's not in the clinical mode. It would be as a usual course of Covishield as Covaxin. So I believe questions are done. We are also overshooting time.
Pankaj, can I ask one question?
Yes. Thank you.
Yeah, there's a lot of apprehension about this vaccine worldwide and skepticism. My question is whether are we using this mRNA technology first time along with the scaffolding lipids nanotechnology? This is a really serious concern that what is wider ramification in the future, in the offsprings and all those things. Could somebody answer on that?
Yes, sir. So the messenger RNA technology, the first question is whether it has been used before. No, sir, it has not been used in commercial stage before. Second is the concern about whether it will be possible to do this. So I told you the main problem is logistics of creating a minus 20 or minus 70 degree temperature cold chain throughout from production to distribution. That's the biggest challenge. Now, this is a very interesting question whether there will be some problems later on, etc. So first, messenger RNA only creates proteins. It does not alter DNA. So to answer whether it will have any problem in offsprings, no, none. That might be a concern with DNA vaccines, that's separate. For messenger RNA vaccines, there was a previous thing that if instead of the COVID messenger RNA, if somebody were to put in some other thing messenger RNA and the scares about that. So I would say that holds for every vaccine. You could be given a shot and you can't see in what is being given to you. So here, sir, it is just same as any other vaccine. We will rely on the fact that if they're saying that it's COVID messenger RNA, S1 spike protein, it is that.
Thank you, Vidi. Thank you very much. It was a really incredible presentation by the team. Thank you very much.
Thank you all and thank you for joining. So stay safe and please join us for the vaccination drive. Thank you so much. Goodbye.